I try to stay from true or false, unless they're really obvious, because people start to second guess themselves. What does he mean by the? You know, and it gets crazy for people. I rather try to stick to facts. I don't try to trick you. I mean, if it looks like it's tricky, it's probably because I just didn't word it right. It wasn't like, I'm going to try to mess you up. I think that's a way of being an academic bully. It's not, you know, there's so much to learn. Let's try to learn what we need to learn, and I'm trying to assess what you're learning. So that's that part. Then I have essays. Usually there's a few short ones and a few long ones, okay? And I usually do it this way. I might say I want everyone to write on question 5, whatever it is. And you all do that. And then I usually have choices. So I mean, I'll probably ask you all to either draw, label, or discuss the cell wall, cell membrane. I think that's a very important concept. It's going to be on the test somewhere. Usually shows up in an essay. Diversity, culturing, metabolism. You know, we've gone through some really nice discussion questions together. So things like that would be good practice. I would be familiar with some of that. You know, I do that so you won't get blindsided. I may ask you some new things, but generally on the essays, besides like maybe 1 or 2 that's mandatory, then if let's say there's 2 essays, 2 short essays, I'll say you have to do this one, and then pick one of the next 5 or something. So that is my idea. There is. I hope you're reviewing everything, but 2 things. One, if you really didn't cover something and I ask you about it, you're not sunk. It's like, oh crap, I didn't get a chance to look at that. But then the flip side of that coin is, well, you have your choice of 5. I'm hoping you're going to do a really good job on the one that you did pick. So, you know, read your answer, think about it a little bit. If you have to start over and stuff, that's fine. So that's the deal of it. I think it's 100 points. If I remember right, I'll check. Whatever the syllabus is exactly what it's going to be. So I sometimes mix up my different classes. Okay, so review on Tuesday. People want to have a review. I never know how to do that, because if I just start reviewing the outline and the outline is up there, some people say, oh, those outlines are so much, they're too much. What do you want? What do you want me to do? Just put up the questions I'm going to ask you? I probably won't do that. So if you don't like the outline, tell me what you do like. Same thing with review. We'll say like, okay, do you have any questions? And people go, no, I've got to get going, catch a cup of coffee. So, you know, I'll try to come up with like some kind of little review, and then if we want to go from there, we'll go from there. I will say that I want to get through part 1 of viruses. I don't know if we'll get there today. We might do it on Tuesday. You go like, oh, I don't have any time on Wednesday to study. Well, then, you know, try to get through that. At least through viral structures is probably about the first third of the chapter. We're not going to get into disease and transmission too much. So I'll try to get about that far between now and Tuesday. So if you want to look ahead, that's fine. That's the idea. Genomics isn't on there. What do you want to know about the test, or what do you want to do about a review? If you have some ideas, I'm here to help you, but you gotta help me too. What?
Okay.
Yeah. Or yes.
So like the quiz questions really help because it like explains how you would format questions, but we only get 10 of like out of the bank. Is there any way that we can get all the questions from like the whole bank of questions so we can like practice?
You want all the questions from the test bank?
No, the quiz.
The quiz?
Yeah. Because like the quiz like bank is maybe like 30 questions and we only get 10 of those. So we don't get to practice on other concepts.
Okay, I never thought of that. Let me think that over. I guess so. The idea was to give you a sampling, and then of course, you know, to give everyone a different set. Let me think about that. We can maybe do that. I'm moving towards but let me think about it a little bit more. So that I don't do anything that I'm going to go, oh crap. It's kind of should be tattooed in the back of my head. Oh crap. Okay, what else? Okay, so stuff comes to you, you know where to find me, write to me, try not to call me on the weekends, but you can if you really want, that kind of stuff. That's all good. Now, this is an interesting lecture to me, and it might not be for you. So, because I have found over the years, people don't like physiology, oh, I'm sorry, go ahead.
I'm sorry. So we have ecology right after this class. What happens if it takes more time in the class?
This is what we've done in the past, and Amy's been okay with it. If people are still writing when class starts, oh, I don't cut off the test for you. I mean, I could, and then everyone goes, you're done. That drives some people absolutely nuts. So I decided not to do that. On the other hand, you know, I've had people sit for 5, 6 hours just looking at the paper, and I said, well, when the sun comes up, you're buying your coffee. So what we've done in the past is we've moved out here to finish things up. I don't think it drives Amy too crazy, but I don't know what else to do about that unless we have suggestions on that. But that way it's like, well, I know I gotta kind of keep moving, but if push gets to shove, I wanna get points, and I can probably catch up in lecture. Do you have another idea?
Also another class, right?
Oh, yeah, and I just don't know, because if it's not reserved, but yeah, of course, if it's more quiet, some people come back and sit in one of the labs, that's fine. I don't like to impose that rule. But on the other hand, I do try to write it so it fits within about an hour, realizing that some people are done in like a half hour, but some people will always sit for a long time, and there's some things I can't control, but that's the way I do it. I had a person last year that took 9 hours on one of my tests, which was, they were eating lunch and talking to people, like, can you sit down and finish your test? So, anyway, but, a deal's a deal. I said, no time limit, there's no time limit, but try to be respectful that, we do have other things to do in our lives, so stay on task. So, let's see if that's okay. If you guys are talking to Amy today, and I'll try to catch her between classes, but I can ask her, but that's been pretty good. It's maybe been 5, 10 minutes. Oh, I just want to reread my question. Okay, cool, that kind of thing. All right. Okay, that's how I do it. If you got a way to improve it, you guys are all super smart. Tell me I'm just a poor professor. Okay, so diversity, metabolic diversity. Now here is a way to start tying together this theme on diversity again. We have size. Right? Size is a big deal because we can put bacteria in different places. In fact, even in a soil, we look at the soil and think, oh, the corn grows in the soil, all that cornfield is about the same. But when you look at it from the microbial perspective, soil is actually an aggregate. And they've even looked at an aggregate, just means a clump. But even if you look at a small clump, a few millimeters, the tiller's going through it, and sometimes they plow it, now they don't, whatever they do. But soil can have an oxygen gradient going from the outside to the inside, actually to the point of where you can have anaerobic conditions going on, even in a soil aggregate, right here in the topsoil, over here in this cornfield over here. You're like, well, wait, there's not supposed to be anaerobic stuff going on. That's in the wetlands, no, you gotta look at the scale. Okay, so I can have denitrification, which is an anaerobic process going on in my cornfield. I'm a farmer. I'm adding ammonium. I'm adding ammonium, which is being turned to nitrate. I don't want to lose that stuff. And it's like, well, you got to think about it. So diversity is a big deal. 1 million billion bacteria in a gram of soil. How can that be, and how can they be so different? So you start asking those questions and getting some insight to that. 1 million billion cats, or any kind of species that fill this floor. Okay, so it's a perspective that you're now getting to appreciate. What else helps us to know about diversity? Okay, well, we're still going to learn. We're going to get this next lesson really pushed into us in the second third of the class, but microbes have been here 3.9, 3.8 billion years, on a Thursday, 2 o'clock in the afternoon, that kind of a thing. They've been here a long time. So you go, so what? Okay, is older better? Well, no. But when you think about the Earth has gone through a lot of different transitions. There was no ozone. We talked about different light being there, and hot, smelly stuff that now they live in geysers. They found places on the Earth that when they evolved, it made sense. When oxygen came through it, was the first big species disaster, knocking out all the species. The first great extinction. We wonder if we're entering another extinction. It certainly comes up a lot with global climate change. So there's a thing to go with diversity, and then we talk about what else could help diversity that we just got through. Growth, generation time. Okay, if you waited for, what did we look at, 10, 20 generation times, you're going, oh yeah, knock that off in 10 hours. 20 generation times with elephants, it's going to be a few centuries, right? It's like they just don't go that fast. Humans go fast enough, but it still takes a bunch of years. So there's things like that that start to compound and do metabolic diversity. So when we see these things today, the thing I want to impress upon you in this prelude to the lecture is that you think about the implications of these functions, maybe how they came about. The concept that nutrient cycling probably wasn't really cycling much until about 2 billion years ago. Well, that's fine for me. I've been here on Earth the last few decades, okay, but it does have different implications as we change the chemical constituency of our environment. Is not a surprise that we're probably changing the natural biome, and we think that maybe the microbes will respond. How many guys have studied or at least are familiar with the Gaia, or some people say Gaia hypothesis or concept? Okay. Well, do you want to hear about it? Okay. It's wild. Okay, if I say Lynn Margulis to you, what comes to mind? Anything? You said, no, nothing. I'm dead there. Lynn Margulis came up with the idea of endosymbiosis. I mean, she really did it. Yeah. So, bing, yeah, the light bulb. So, what is endosymbiosis? I mean, in a nutshell. Give me something on that. You guys know this. You learned this in high school now.
It's like some of the organelles in eukaryotic cells, like mitochondria and chloroplast, were theorized so that they came in by small microbes just got engulfed in the cell.
Yeah, in a pre-eukaryotic cell, we had this engulfing. Well said. Good job. Supposedly a proteobacteria was engulfed but not digested. Okay, and over the years, a lot of the material went away, but the energy making part of it, the citric acid cycle and so forth, really was localized, and the membrane thing, that wasn't a problem. They doubled that, and we have these wonderful power packs called mitochondria. Cyanobacteria, actually, the lineage turns out right before the cyanobacteria, but close enough for us, came in and became the chloroplast. Wow. Organelles that can take light and make little chemical batteries. More inspection. It seems like maybe the eukaryotic flagella may have came from spirochetes. The same kind of things that cause Lyme disease. That kind of stuff. Really? Well, they're still working on that one, and there's a lot of discussion about the nucleus. Some people think the nucleus now comes from the planktomycetes because they actually have kind of an internal organization. Okay, and some people say, well, the nucleus could be a virus. And really still trying to figure out how viruses all fit with everything. They're kind of an interesting thing. But this idea that cells can that we are really an amalgamation of little bacteria pieces over the year kind of blew people away. People thought, what a bunch of hooey. And when it was first presented, endosymbiosis, there's some people that think about this, we don't know, and in the 60s and stuff, nah, these people are high, they don't know what they're taught, it's nothing. And then they started getting into molecular biology, so wait a minute, the nucleic acid in that mitochondria kind of matches up with some of these proteobacteria over here. When you look at the tree of life, they say mitochondria. You go, why is that there? Oh, that's where it kind of fits. And in the chloroplast, oh, that fits over here in the tree. And if we give these things antibiotics, there's a response. And they start doing all this stuff. They're going, we thought Lynn Margulis was nuts as a whatever, crazy gal. But she was right, okay, she was right. I had the privilege of teaching with her for like an hour, or actually she taught, I listened, but, you know, she's a great person. Well, to get back on point, so that was tying something, hopefully you know. Margulis, with a guy named, it's like Lovelock, Lovejoy, something like this, something like love. They come up with this idea called the Gia hypothesis. I'm not probably saying it right, I never did know how to say it, but you can Google it, it's out there. The Gia, I guess, is the goddess of the earth, and that's why the name. The idea with them is that the earth is like a super organism, you know, and that it will regulate its own climate, it will regulate where, you know, what it's doing. And they said, at some point, you know, people say, well, that's that's crazy, you know, batshit logic, you know. But then they go like, but you know, she was right before. You know, so that's always comes up. But then they argue things like, well, why is the atmosphere of the earth, why did it stop at whatever, you know, 18% oxygen? I mean, you know, we could produce more, but there's some kind of regulation mechanism, realizing that the microbes changed it from, you know, 0 to that in a matter of, you know, eons, okay, you know, and they start talking about some of these different arguments, and it's an interesting hypothesis. You should look it up just as like, you know, you bring it up and argue with it, you know, to some professor or something, and see what they say. But it should be something you're aware of. Is it the case? Maybe. The whole thing for us, in our extinction, in our diversity, really, the hinge pin is timing. You know, do we have enough time to evolve? If the microbes are regulating everything, you know, are we giving them too much to work with? You know, that kind of thing. So, you know, no one really knows. But when we start thinking about diversity, start thinking about, you know, different themes we've talked about on that. Let's talk about metabolic diversity. You know, some stuff we know about photosynthesis. We know what that is. Phototrophs carry out photosynthesis. Most phototrophs are autotrophs. Why would they have to say that? What does that tell us if it's an autotroph, just as starting to review? I know we've talked about it. If something's an autotroph. Go ahead.
They use CO2 as a carbon source?
Yeah, carbon dioxide, so that's good. We have to have pigments, you know. We're kind of stuck in this idea initially that pigments are green, but this time of year is kind of cool because the green goes away and oh, there's other pigments in there. And then we start looking at the bacteria and oh, there's other pigments there, and you know, other things can collect light. We kind of get that as biologists. So, and we know what photosynthesis is for. That's not a problem. But you know, we need to open up our ideas here. This is the photosynthesis we're all know about, right? So that part of the tree. This is a new one that you can actually use organic carbon instead of CO2, and still use that organic carbon to capture, to change the oxidation state and capture some chemical energies. So that's a little different. That's something you probably didn't pick up in other classes. So if you see these words together, you know, it means something. Heterotroph really is going back to organic. You probably learned it as, well, heterotrophs are things that eat other things. Well, you know, in a way, yeah. It's just that so they're talking about a more complex organic carbon. Okay, if we look at the phototrophs, you know, they don't always have to be oxygenic. We're pretty used to energy coming in, or CO2, and then we get reducing power off of water that goes on to the CO2, makes a carbohydrate of some sort. We most know that N is 6, and we're all cool with that, right? Or you should be. We have an electron shuttle. We have these different photosynthetic areas that capture light. We know there's a photosynthesis 1 and 2, and we know that 1 is cyclic, and that cyclic photosynthesis involved in the cyanobacteria is maybe something that you may or may not know. When we get into some of the ones you're seeing in lab, and if you're not in lab, we took mud, we had some sulfur, we had a little bit of carbon, well, I think that's about it. Maybe some people had nutrients or something. We put it in the window, and just the light in mud. All of a sudden, we were seeing purple bands we saw the other day, and yellow bands, and there's always some green stuff. You can have you can have a type of photosynthetic reaction going on without having the generation of oxygen. Okay, so before we talked about something being anaerobic because we were using oxygen as a terminal electron acceptor. Here, the key word is genic. Okay, we know from photosynthesis that we generate oxygen. We're all pretty grateful for that. But you can, so you're going from oxygen that's reduced to oxygen that's oxidized. Easy for us to see that. If you have another fairly electronegative compound, sulfur, and you use that to harvest the energy off of the electrons off that, okay, you can oxidize it. We talked about that. Oh, some organisms save sulfur. And you can oxidize it even further if you have the metabolic machinery. So, for us, again, this sometimes throws us, oh, oxygen is good. No, I mean, oxygen is oxygen. It's pretty cool stuff. But it can happen without that. So a lot of us are thinking with the Winograssi compound, that's so weird. You put an oxygenated sulfur on the bottom, and why did you do that? Well, the thing is that you'll have some organisms that start out and use the oxygen as a electron acceptor. They'll reduce it, and as they reduce it, then other ones will come in with their pigments, pick up the product, and then use it and oxidize it, and that's what sets up that window, that gradient that we can see with our eyes. Some of us will take apart these photosynthetic columns and pull some of these guys out and take a closer look at them. They're kind of fun to watch. Very, very interesting. Okay, so photosynthetic, we know of chlorophyll. We have, two big energy centers. It is not them. I think in general they make you memorize these and the numbers and stuff. That's not really my point here. You have that lesson. I'm okay with that. I want you to be more aware of that with bacteria chlorophylls. Okay, there's like a bacteria rhodopsin in Halobacterium, a halophile. You always see these pictures of these salt flats, and they always the authors always say, look, the salt flats are red, and we're all supposed to go, ooh. But if you don't know that halophile bacteria convert from heterotrophism to a light reaction and make bacteria rhodopsin, it doesn't make a lot of sense to you, but that's what they'll do. They'll try to get light energy in the end if they're running out of other energy. You know rhodopsin as a light pigment, right? What's the trick you do with rhodopsin, or some of you? Do you know that one? Maybe you ever try to sneeze? You want to do anything to try to stop, to make your sneeze go? Look at that. Don't look at the sun. But you look at the lighthouse. Rhodopsin goes ding, and it sends out a signal, and you're like, oh, I got to balance my CO2. Achoo, and then you're there. So you're right on the edge of trying to get the pH in your blood back to normal, so you're sneezing. So, or yawning. So, but there's other different wavelengths that then therefore, where absorption is going on and where light is being reflected. We see green because blues and oranges are mainly being used for light. We see purple because some of the some of the bacteria, some of the higher wavelengths are being absorbed, and purple is being reflected to us, and that's cool. So what's the take home? That the spectrum is wide, and if you have diversity, different organisms evolve to harvest different parts of the spectrum. It is likely, I used to have this discussion with some of the other professors, it is likely that maybe green became the Earth because the other wavelengths were occupied or being used, and there was an advantage to go with harvesting and reflecting green. I heard this actually from a botanist who we talked about pigments one day. I don't know how to take that argument too much further than that, but it's something to think about, should you like to. Nothing, again, more review. We know that there's other pigments that help with things, help with gathering lights. In the algae, we see it a lot more, these auxiliary pigments that help with it. Not gonna go into too much detail in here. I think in general, do you still do the acetone and suck the different pigments up and look at the bands and stuff? Anyone, okay, so you guys got that lesson, that's cool. So. Now, antioxidant photosynthesis, 5 phyla. Now, these phyla may or may not mean a lot to you, but let's start kind of looking at them, because we're gonna hit the phyla really hard. There's about 39 of them, which just is amazing, 39 phyla. Okay, one that you should know are in the proteobacteria. Jake and I were looking at the Winograsky tons. Oh, there's purple ones. Purple sulfur bacteria are a beautiful one. They're green sulfurs, look just like cyanobacteria, but they happen to use sulfur. And then there's also green non-sulfur, those that will be green bacteria, not cyanos, that are using heterotrophic force. And some of the gram-positives will do it. So for now, do you have to know that? No, the point is 5 out of 39 or so phyla have it. It's just not incidental. It's co-evolved. There's different things thought, oh yeah, getting energy from light is a cool idea. So what they're doing, we know about. We learned the other way. We learned catabolism. The movement of electrons. Those of you who've gone through and learned photosynthesis can recall that you're moving electrons, but in the opposite way to reduce carbon. And so that still is by the same basic mechanisms we talked about. So some examples here. Photosynthesis in bacteria has been studied in membrane systems. Oh, I know what I'm talking about. Yeah. So what? Somebody say something? Oh, you are? Just throw things. Shut up. Okay. Anyway, chromatophores and rhodobacter. Now, rhodobacter is a purple sulfur. And the vesicular photosynthetic membranes are continuous to the cell membranes. So, you see these little pock marks in here? And we tend to think, oh, bacteria, they're little circles, they turn pink, they turn purple, that's kind of it. Sometimes they're kind of squiggly shaped. But the deal is that these pock marks are the membranes that have been pushed in. So you're looking at the pushing in of the membranes either from the top down, because this is an EM, or the bottom up. And so what are you doing when you kind of in pocket or you start to fold things? Like the villi of your intestine, you're increasing surface area. Okay, you're increasing the potential to do proton motive force by doing that. And so sometimes you need that by creating invaginations. Another way, and this is a cool one, we'll see this several different places, but here in the Ectorhodospira, we have this folding of these membranes. Now, if you just took this class, and or didn't take this class, and I put this on a test, you probably would guess, well, I don't know, it's got to be some kind of eukaryotic cell. Look at all that membrane folding in there, and maybe that looks like, what, a Golgi apparatus, an endoplasmic reticulum? I'm not sure what you got there. Well, what it is, it's truly a bacterium. There is no nucleus. It is kind of hard with these EMs to interpret them sometimes. But this kind of situation is the membrane coming in and folding back and forth on it, and you're kind of catching it with the EM. It might be folded in a U or something, so you miss parts of it. It's hard. It's hard to get these pictures. But there, you're definitely increasing the membrane potential. And where we start to see these things is when you really, really have to move electrons around, and it goes against the grain of things. Okay, when you're trying to oxidize methane, when you're trying to oxidize ammonia and some of those things that are just really reduced, it takes a lot of energy. The cell has to get a little bit more of that battery thing going for them. So this is neat. I mean, to me, I don't know. It's like, I'm gonna tweet my friend. Oh, what are you doing after class? Okay, but I mean, I think this kind of thing is, just helps to show some diversity, and again, its structure being coupled with function, so that type of thing, again, is part of the diversity. Not an organelle, but still an adaptation. Okay. Wait, I don't wanna say too much about these things. Oh yeah, so you got a photocenter, this is just to say, it's about the same kind of thing in the membrane. You have a different type of photocenter, okay, you have, movement of protons on the outside, and then eventually, production of ATP. That's about it, these kind of photosynthetic reactions are going on in the membrane. I don't need for you to memorize this, just like I don't really need for you to memorize the cytochrome system in your cells. I think we can talk about them with pictures. But we have some of the same kind of things going on, in this, that in anisotropic photosynthesis, reducing power for CO2 comes from the presence of reduced compounds. Even nitrite in itself is a little bit more reduced than nitrate. Requires reverse electron transport for NADH production in purple phototrophs. So basically you're moving the electrons in an opposite direction, which is really what is going on in photosynthesis. So it's just the point here is that you're working by the same rules, but you're just using a little variant on the photosynthetic idea. And quite likely this evolved before photosynthesis, because this is cyclic photophosphorylation. Okay, what is it in photosynthesis 2, then photosynthesis 1, it's non-cyclic, it moves down the chain. Cyclic photosynthesis came by first, so it's less efficient, but it still gets the job done. Probably know a little bit about that. So chemolithotrophy, we're just going to do a little dance through this metabolism. Chemolithotrophy, obtain energy from oxidation of inorganic compounds. Obtain energy from oxidation. So this is an energy thing, okay? Chemolithotrophy. Litho means rock, but that's your inorganic component. Okay, the chemo part is the energy component. Carbon is a different story for these guys. Okay, oxidized inorganic compounds, but use organic carbon as a carbon source. So let's take a look at a few examples on that. So here we're not worried about photosynthetic pigments. Carbon dioxide can be the sole carbon source. And electron donors can include sulfide, hydrogen sulfide, ammonia, nitrite, those kinds of compounds. Well, there's a contradiction we should talk about. I didn't see it till right now. Some can use organic carbon. Some can use carbon dioxide as the sole carbon source. That is true. The point is, chemolithotrophy usually uses carbon dioxide. The mixotrophs, which we're finding to be more abundant, can use the organic. So that's a term you might come across, mixotroph, because they're mixing the carbon source with the inorganic compound oxidation. So it's not a contradiction, it's a subset, right, of chemolithotrophy. Okay, oxidation of reduced sulfur compounds. Many reduced sulfur compounds are used as electron donors. And we've talked about, as long as you can oxidize it more, you have energy potential. Okay, oxidation potential of sulfur, we have like 7 different common types, I think, 5 to 7, I got it. So there's a lot of room to have steps, and that worked out really nice for things. So here we have hydrogen sulfide going through a system, that's all right, and we end up with sulfate. Sometimes it only takes a part of the way, but enough energy can be produced there to reduce things and still make up some electron transport and energy production. In the end, you can get sulfate from different portions. The point of this is not to memorize this part. The point is that until you take physiology, the point is some bacteria will do this. Okay, we'll go straight to sulfate. Some ones we talked about may stop at sulfite or elemental sulfur, but then later on they store the sulfur, they can use it later. Okay, so the take-home message is in having different pathways embodied in different bacteria, that too helps have species diversity. Because you have some conditions that are really anoxic, and some things can be adapted to that, where some other situations aren't quite that reductive, and having an intermediate is important. We'll talk about syntrophic relationships too. Sometimes you have the ability to do most of the pathway, but you don't have part of the substrate to start it, and you might need, you know, your buddy species to help you along, and that, we'll get that in a little bit. We've kind of covered this idea, the idea of oxidation of reduced sulfur compounds. Basically sulfide eventually making the sulfite, moving electrons mainly down one direction, and in some cases moving them backwards to get back onto the electron shuttle and recycle NADH. Again, I show this mainly because it's what, fermentation or respiration? Gotta be a respiration process. Is it anoxic or oxic?
Toxic?
Yeah, you're right, actually, that down here, yeah, this is an oxygen one. Let me look at this. Yeah, so we have oxygen as a terminal electron and stuff in there, and, yeah, oxygen is listed in the second point, yeah, good point. I was thinking of something else, good. Okay, nitrification, ammonia and nitre are oxidized by nitrifying bacteria. We bring this one up a lot because we use so much nitrogen in Illinois that the nitrogen cycles are a really big deal to us. Nitrification is a big problem for some of the farming industry. People put ammonium what? Hydroxide. It's a gas, they inject it into the soil. And it's a good idea, you know, because a soil has a very high cation exchange capacity, and so it likes to grab the positive ammonium, or ammonia. The problem is nitrification, okay? The microbes, and they even put inhibitors for the nitrifying bacteria. They put in nitropyrene and other things to stop microbes from oxidizing the ammonia. But the problem is that they still do, and it contributes to Gulf hypoxia because the ammonia turns to nitrate, and nitrate is water soluble, and then it goes down to the Gulf and other places and causes Gulf hypoxia. Did you know what that is? You should know. You probably know. You know what Gulf hypoxia is? Nobody? Well, let me tell you another story. I'm trying to get too far off on this, but it has to do with nutrients and bacteria and agriculture, which is kind of like our lives in Illinois. So we just had the story of ammonium going on, oxidizing. Now the nitrate is soluble. That goes into the drainage ditches and into the streams, and some of it affects us locally. And if it's too high, it can cause disease. Not really linked to cancer, but things like blue baby syndrome, things like that. Methylhemoglobia, it's a super long name. But also, not only Illinois, but Illinois is right up there, being in the middle of the Corn Belt. These high nitrate waters make their way down to the Gulf of Mexico. You guys might. I have to talk to Amy. You should know about Gulf hypoxia. So, the nutrients make it to the Gulf, okay? They go past New Orleans, hang on Bourbon Street for a little bit, get some crawdads, but then they go into the Gulf. And there, nutrients hit a whole bunch of algae and phosphorus. And what happens to the algae, do you think? Anyone know if I put a bunch of nutrients on plants, what do? They grow, so you have this big bloom, and what happens with algae after they bloom? They don't live like redwoods, right? They die, what happens then? Yeah, they go down, they decompose. Decomposition does what to oxygen? Uses it up, there you go. So hypoxia doesn't mean anoxia. but it's pretty close. So your crabs, your fish, you know, all these things that are used to hanging around in the Gulf are having a tough time. The hypoxic problem is a national concern. I haven't been up to date on this year, but it's roughly the size of the state of Massachusetts, which is not a small state. I mean, it's smaller than us, you know, we're better in Illinois, we're bigger, but, you know, it's a big area, okay? So hypoxia is a deal. Now, back to the nitrifying bacteria. Nitrifying is kind of tough. You got this really reduced chemical, as you all know from chemistry class, and you need to oxidize it into two stages. So let's get into that. We have enzymes that make sense to us, ammonia monooxygenase, oh I see, and then there's several different steps in this thing, but the energy release of this is variable. We'll talk about it here. So in nitrification, I know this, step one, you should know that nitrification then is moving ammonia to through ammonia hydroxide, but then when the first step of nitrification moves ammonia to nitrite. Okay, this is a big energetic step. Okay, it's very, very hard to do it. You do it in the presence of oxygen. It's not an anaerobic process at all, and if you look in the back of the book at the kinetics, the amount of power it takes to do this, it's really high, okay? Because, you know, you're pulling off, you know, so many hydrogens and adding even at the end, 2 oxygens, okay, to get to this step. Several enzymes involved, okay? That's kind of the rate limiting step, you know, and people say, well, how come I don't see a lot of nitrite in the environment? In fact, if you see a lot of nitrite in the environment, it's usually like at the end of a sewage treatment plant or something. It doesn't really exist a lot because when you move from nitrification into the movement of nitrite to nitrate, you can see the reduction potential difference in here is next to nothing. So usually in nature, once you move to nitrite, something the second step of the nitrifying bacteria come in and bang, nail it and oxidize it further. But that first part is crazy. The first bacteria that do the first part of it have that big invagination membrane. You know, they need that extra potential to oxidize ammonia. We'll see it again when we oxidize methane. Okay, with the methanotrophs, the guys that like to eat methane, something that doesn't sound terribly appealing to us, but I suppose nitrification doesn't either. What do I have? Fixing nitrogen. Okay, so fixing nitrogen is a different current. Does anyone know about nitrogen fixation? About half of your crops here do nitrogen fixation. The ones that aren't corn. What are the other ones? Soybeans, yeah, they're amongst them. Other legumes, alfalfa, clovers, you know, they fix nitrogen. Okay, again, it's not that like I'm fixing a flat tire kind of thing. It's they're able to take nitrogen in gaseous form, which is one of the most abundant forms of nitrogen, and reduce it. Usually down to glutamine or a different amino acid. To do that, you have to reduce it, okay? And to do that, it doesn't work very well with oxygen around, so some of you may know this. Soybeans have these nodules, different legumes have nodules. What happens is, to have this go, is that the plant will actually be putting out quinines, will be putting out different chemicals that attract 2 different types of bacteria. Bacteria that will come and help to take a root hair and turn it into a nodule, but then also try to bring in the bacteria that do fixation. Okay. The nodule is an interesting thing in that it will allow enough gas to get in but reduce the oxygen enough so that you can reduce nitrogen, which is, two ends in a triple bond. It's a fairly stable compound. Lucky for us, that we can light a match and not blow up ourselves with gaseous nitrogen. Cyanobacteria do this also. And they have a special, instead of a nodule, they have a special structure called a heterocyst. And the heterocyst is leaky enough to bring in gases, but can reduce the oxygen tension enough to allow nitrogen reductase, maybe it's on the next slide, to reduce that. So that's a pretty cool thing. If we could do that, and it costs a lot of energy. If we could do that, it would be a neat thing for us, but it does take a fairly big energy input. So, energetic redox considerations. We've talked about respiration. We've talked about fermentation. In the absence of external electron acceptors, compounds are fermented. This was the basis of that question 3, remember? So, fermentation's okay. We'll go to it. But if there's an electron acceptor in there and you got the mechanism, you're going to go for that. And that's what that E. coli was doing, right? And then you add oxygen. It's like, well, now I'm going to choose between the not so good electron acceptor or the okay one, but now I got the super electron acceptor, and I'm going to go for that. So when you, this helps you to understand when you're learning about nutrient cycles, right? When you're thinking about when nitrogen works and where. If we have oxygen in the environment, we're going down either in depth, in water or soil. We have then, depending upon the conditions of the soil or the water, we have reducing our oxygenic situations. Usually there's something here producing oxygen, there's algae in the soil, algae in the water. But we know that we're starting out here with what, about 200 millivolt potential with oxygen, and if we take it all the way to the most reducing circumstances, it's minus 400. Okay. And then, so here, when we look at the Earth, near the surface and with air, oxygen's our first electron acceptor. When we start moving down, some of the metals work, okay, and then we start getting into nitrate, sulfate, and then finally carbon dioxide. Carbon dioxide is terribly hard to reduce, and you really have to have the conditions around that. You don't see carbon dioxide as an electron acceptor too much, because because of the nature of carbon versus the nature of sulfur and nitrogen. So probably a review from what you've learned in other classes, but it kind of ties together when and where some of these things are going on, both in space, both in the cell and in the space of Earth or your gut or in other places. So, you know all that. Okay, so energetic redox potentials. We go into looking at pyruvate. We're going in and looking at hydrogen production. Involves the transfer of electrons. You know that to acetyl-CoA. And we have acetate. If we can have other pathways coming off of pyruvate in fermentation. So here we want to think about fermentation for a second and realize, and this will be important in lab, fermentation isn't just always produce one product. It sometimes produce 2 products. And what it produces with it is also important. Okay. To some sense, you know it. You can see that you get alcohol and CO2 from one pathway, and another pathway you see, oh, I only get lactic acid. So in some ways, we're restating what we already know here, but we're going to use that to our advantage when we get. It's the next slide. When we get into studying bacteria. And then trying to use chemicals to identify them, which is what we do in lab. This is, glucose produces a single fermentation product, so in homofermentation of glucose, we get a single product in the end, lactate. There's many things, glycolysis, substrate level phosphorylation going on here. We get to pyruvate and then to lactose. So this is called homofermentative breakdown of glucose. And in heterofermentative, this is an example where we get ethanol and carbon dioxide production, carbon dioxide and ethanol. It just means that you're getting mixed products coming off of the fermentation process. That's it for our purposes. In the when we're doing this in lab, it's very important to us if we have bacteria that produce acid and gas as opposed to not produce acid and gas. And that becomes a way of separating things like the enteric bacteria. So whether they're doing a mixed acid fermentation or a straight fermentation becomes important. I want, hang on for a second, where is, oh, here's, it's coming up. So, we're in the, a variant of the glycolytic pathway, the antidiuretic pathway, we're simply taking sugar and breaking it down in different ways. The important thing here is you may see this word, we know different ways, but what's the take-home message? In mixed acid fermentation, we generate acetic acid, lactic acid, succinic acid as a result. This is an easy way of, did I go over the time? Okay, good. I'm up here in my own little world. So in mixed acid fermentation, like something like E. coli does, we can look and see the acid. It's an easy thing for us to measure, pH, or we'll use color to do it. In those that don't do mixed acid fermentation, we look for other products. So here we have glucose going through glycolysis. Okay, if we have mixed acid fermentation, that takes us one way for acid. If we keep going down for another group, and here here's another genus. This is the point here is these are two different enteric bacteria. If some of us may recognize coli, I think you recognize coli. This is its cousin. Okay, the difference though that distinguishes this genus from this genus, even though they're in the same phylum, is that there's a breakdown product that goes past pyruvate, and then is eventually going to make a breakdown product called 2,3-butane-diol. If nothing else here, in this fermentation pathway, if you can write down acetone and 2,3-butane-diol as a product of one of the fermentation paths, we're going to see that again. That's a diagnostic characteristic of like half of the enterics. The other half, more or less, are kind of following down this other pathway. And we'll see how, Proteus, coli, and stuff fit together, but Enterobacter, Salmonella, some of these other ones. Why do we care about this product? Usually we just care about, this intermediate, usually we just care about the product. The thing is that it just happens that we can do an easy indicator in the lab to detect acetone. Okay, we do an easy indicator in the lab to look for nitrite when we look for nitrate reduction. Acetone is our indicator organism as opposed to looking for acid. Okay, syntrophy. Syntrophy is interesting. Once I explain it to you, you're going to get it going, yeah, I get that. But then the problem I've always had is once I think about syntrophy, how do you know that everything isn't syntrophic? And it's kind of like that argument you have about parasitism. When is something preying on something, and when is something being a parasite? You just have to look at the strength of the relationship. But we're going to keep it real simple for our class. Maybe we'll discuss it more. It's like that discussion, are viruses really dead? Okay, can we just, agree? So syntrophy. a process where two or more microbes cooperate to degrade a substance neither can degrade alone. Okay, that's the key, is that you're looking at the original substrate, as opposed to, I'm bacteria A, I spit out, I don't know, sulfur, and the other guy says, oh, I want that sulfur, I'm gonna take it and do something else with it. That's kind of a cycle. Syntrophy is looking at that original substrate. So most syntrophic reactions are secondary fermentations, okay, they're happening after something else. Most reactions are based on interspecies hydrogen transfer. These at least are the most famous one. Hydrogen production by one partner is linked to hydrogen consumption by the other. A little premature for this class, but hydrogen in some cases shuts down the reactions of other organisms. You think, oh, great electrons, why not do it? But it can also have the effect of gumming up somebody else's respiration system. So having an organism around that can suck away or take away the hydrogen that's being involved frees up the original organism and benefits the second one. That's the benefit thing going on, why it's syntrophic. Syntrophic reactions are important in anoxic portion of the carbon cycle. We see it with the C1 cycle, one carbon cycle. Hydrogen consumption affects the energetics of the reactions carried out by the hydrogen producer, allowing the reaction to be exothermic. That's not as important for us. Here's our example, ethanol fermentation. Okay, so you produce ethanol, 2 carbons and a hydroxide. Okay, got water, and from that you get hydrogen, some acid, and then acetate. Okay, so in methanogenesis, methanogenesis, this means the production of methane, genesis, and this is happening by an archaea. Okay, you have hydrogen, which you wouldn't know, but they're the only ones that really do that. Hydrogen plus CO2 gives us methane. Okay, the couple, so one is producing the hydrogen that goes over and can be used for methanogenesis. That's the key, degrading the alcohol. A couple of reactions, so methanol plus CO2 gives methane and acetate, which couples the two reactions, and therefore they are linked. So, and the big thing that links them is the hydrogen energy transfer, which the fermentators can't do. Okay, so syntrophy is that linking. I don't think I would just put reactions up there for you, but we can at this point talk about, when we bring up syntrophy, we'll bring it up again when we study the archaea. It's a metabolic process that is different in that the full process isn't in any one bacteria. General principles, I think we've covered all this stuff. This is just for you. We've talked about this quite a bit, the electron transfer, or electron tower, and I did want to cover this. I don't think we've covered assimilative and dissimilative. I don't know if you cover this in your other physiology class. In assimilative metabolism, but you know, when you look at the nutrient cycles, some are assimilatory, some are dissimilatory. In the assimilative metabolism of an inorganic compound, the reduced compounds are used for biosynthesis. So it's easy to remember. It's assimilated. It's used. Okay, that's the deal. Sometimes, if the compound is just being used to give away reduction potential in the form of electrons, you know, or if it's just being used as an electron acceptor, you can change the oxidation state, but nothing's getting assimilated. Okay, this is really important to us because this is how nutrients are cycled in the environment. So during anaerobic respiration, the reduction of an inorganic compound is called dissimilative because the reduced products would be excreted, not made into an amine, an amino acid, or glucose or something else. Okay. So those are terms that are important in microbiology. Oh, okay, that's the end. You have any questions? Take home, go through and look at the definitions, look through the examples. I'm not gonna ask you to write the chemical stuff, but see what's going on. Can you recognize this? Can you connect some of those terms in this sampling on that? Yeah. Stretching. Okay. Okay. 7 minutes. You want to get started on viruses, or are you guys spent? I mean, if we start today, it's less we have to do on Tuesdays. So I do want to get to the first part of viruses. What do you feel like doing? Go for it? Yeah. Yay. Okay. Love your stuff. Actually, viruses are a lot more fun. Okay, one thing about viruses is we all have kind of an idea of viruses. I mean, it captures your imagination. I never saw this one, but, we have other things. The hot zone, anyone ever read that? George Preston. I had a bunch of them. I don't like him, but he's a good writer. Okay? But I'm sure he doesn't like me, okay? But we didn't get along too well, but that's the way it goes. But we were teaching out east, and you meet all kinds of cool people. We were teaching about microbes. This is like about Ebola, and there's things in there where people's eyeballs are falling out and bleeding, and it's great journalism, and it's more or less based on stuff, just like Outbreak, now an old picture, but you can still go to Netflix and look it up, and they talk about, it's one of these what-if things. What if the monkey bites you and, and spreads to your family and the whole world? It's like a Tom Clancy, novel, the Japanese are coming to kill us, we gotta take care of whatever. I'm listening to a Tom Clancy novel now. So I don't know, I think the Japanese are just great people. I loved it over there. So anyway, we know about viruses. We confuse it with computer viruses, and yet, just like where we started the lecture, sometimes there are some similarities. When we look across disciplines, viruses are kind of small. They do kind of go into random places and cause havoc, but we're going to stick to biology. But we like thinking about viruses. So let's start thinking about them. Viruses are complex. Viruses are small. If I said to you, give me a rough dimension of a bacterium, you'd say it's about what size? No idea. Anyone have an idea how big bacteria are? Yeah, most 1 to 5, but yeah, we can get some bigger ones. We did talk. That's a good point. But most we're talking, and that's even to us. Oh wait, I look at my hair, and that's maybe 70 to 100 micron. It's 100 times smaller than the width of my hair. It's hard to understand. These guys are down 1/10 of the size of that, and just looking at these micrographs with the cartoons above them, give us some idea of what we're up against. The tobacco mosaic virus, okay, a spiral of something. We're going to call those cap smears. That's going to be one of the first things we're going to learn, and it's up there. They make a coating called the capsid. Bacteria don't have capsids. Capsids are protein, or cap smears. They're little bits of peptide that they repeat, because they're just not fancy. They are the efficiency of making stuff. So the spiral is a good thing. This icosahedron, 20-sided thing, it's not like that in nature. No one does. Yeah, that is a real shape, icosahedron. Okay, and if you actually start doing the math on it, it's very, very efficient, and it works really well with taking monomers and putting them together in a capsid form. So it does exist. There it is in a adenovirus. Okay, we have the flu virus. Pisses us off, right? Mutates all the time. Kills half of the United States people in 1980, or 1/3. Could kill us all again. We don't know. Viruses, you don't know. But we see that this one has a capsid there, and it's coated by an envelope. Now, in some ways, like the molecular or the cell wall, the envelope that we talked about, but with the gram negatives. But you know what? Not exactly. Okay, that envelope can come from the nucleus of your cell, a cell. It can come from the cell membrane. And it can help to mask the virus by putting little spikes and receptors and things that make your cell go like, your immune system's going like, okay, that's a cell of you, that's a cell of you, I don't know about that one. And then it's going to send over some stuff and start messing with the ones it's not sure about. Well, it's going through and goes, that one looks like, that's one of them. On the outside, it's like the wolf in sheep's clothing, there's a virus on the inside. And we have viruses, so plant, the most famous and the oldest one, okay, a naked virus, animal virus, and then this one, the little lunar lander. And we go, oh, that's cute. They're not really like that, right? They're making that stuff up. I almost said they made that shit up, but no, I didn't say that. So anyway, they have it. Well, we see some things that are saying, here's that naked virus, that icosahedron with that DNA inside of it in this case. But this one has a tail sheath, and it has some plates and some fibers. This thing can actually go in and find a receptor, dissolve the cell, and take a little squat and put the DNA straight into the virus. These things, into the bacteria. Phages kill only other bacteria. Okay. So you think, that's scary, but then people started thinking before in Russia, they started saying, they're species specific and they'll kill bacteria. Why don't we use these things to fight disease? And they said, yeah, comrade, great idea. And so they started working on it, and the war came in and gutted the science program of the Soviet Union, and every place here too. We were trying to either survive or kill other people, and it was an expensive job. After the World War, they stopped looking at it, but you know what? I looked up yesterday, I said, I wonder if there's a phage that's going to take out one of the top bacterial diseases of today, which is MRSA, yeah. Wouldn't that be interesting if we could just, swallow some phage and well, there is a phage that they're developing for it. But there's problems with it. We'll come up and talk about it. Well, isn't that interesting that we could just, listeria in lunch meat. They want to spray your lunch meat with viruses to kill listeria. I mean, would you drink viruses? I don't know. So the size thing is amazing. The scaling, we talked about that. Love that picture. And we talked about size a little bit. What we want to do here is, in our last 2 minutes together, for a nice weekend coming up, get some terms down. Virus, genetic element. Okay, viruses can contain DNA or RNA, but not both. They can manufacture some RNA in some cases. Okay, the other thing in this, and we're going to have some slides that say this again, but some viruses have double-stranded DNA, some have only single-stranded DNA, some have double RNA, some have single-stranded RNA. The other thing, if you're still going through that molecular portion, try to do that, because some viruses, they have plus-strand RNA and negative-strand RNA. Does anyone know there's any plus and negative RNA? Is that anything you've talked about anywhere else? And I'm happy to help if no. Anyone know? Okay, when you look at DNA, you know it's complementary and anti-parallel, and one part will do transcription to RNA, and you'll get messenger. If you can take messenger and go into make an amino acid and read it, that's the plus sense. Okay, the opposite part of DNA makes a negative thing. You have to produce plus or sense RNA to do translation. So that's important to know. Virology, the study, virus particle, if you don't know what you're talking about, call it a particle. Virion, the infectious viral particle, nucleic acid genome. Virioid, the small RNA molecule. This is not a virus we're going to come into. These are just like little pieces of molecules that come and mess with us. Phage, those that kill bacteria. Take a little look at this. The slides will be up there. We're going to try to get into viral replication and the structure, and that'll be about it. But if you got a little time this weekend, go with that. I don't want to really drop a bunch of stuff for you on the last minute, but I'll talk about those slides, and then we'll do some kind of review, I guess. And go from there. All right, I'll see some of you guys.